Welcome back to my channel Knowledge Chronicle.
Have you ever wondered how scientists grow thousands of identical plants from just a tiny leaf or stem in a lab? Can a single cell really grow into an entire plant? Welcome to the fascinating world of tissue culture, a revolutionary method where science and nature join hands to transform agriculture and plant biotechnology. Stay with me till the end as we uncover the secrets of how plants are cloned, how diseases are removed at the cellular level, and how potatoes and tomatoes were once fused to create a bizarre plant called Pomato. What is tissue culture? Let's start with the basics. Tissue culture is a technique where small parts of a plant, like a piece of leaf, root, or stem, called explants, are taken and grown in sterile lab conditions inside test tubes or petri dishes. These explants are placed on a nutrient-rich medium, which acts like a perfect diet for the plant cells. This method became popular in the 1950s, when scientists discovered a magical ability in plants called totipotency. Totipotency means that every single plant cell has the potential to grow into a complete whole plant, just like a seed does. And thanks to this discovery, we now have a faster, more efficient alternative to traditional breeding. What do plants need to grow in labs? The nutrient medium used in tissue culture isn't just any soil or fertilizer. It's actually a carefully balanced mixture of a few key things. First, a carbon source, usually sucrose, which is just common table sugar, and it gives energy. Next, inorganic salts, like nitrogen, potassium, and phosphorus, to build plant tissue. Then, vitamins and amino acids, to support cell metabolism. And finally, growth regulators, like auxins and cytokinins. Auxins promote root development, while cytokinins encourage shoot formation. The right balance of these hormones determines whether the plant will grow shoots, roots, or form callus tissue, which is basically a mass of undifferentiated cells. What is micropropagation? Now comes the best part. When we use tissue culture to produce thousands of identical plants in a short period of time, this process is called micropropagation. Each plant grown this way is genetically identical to the parent plant and is called a somoclone. This technique is used on a large scale to grow banana, apple, grapes, tomato, and many more crops that are in high demand and need disease-free, fast-growing varieties. Imagine turning a single banana plant into an entire banana farm, all with identical fruits and qualities, thanks to micropropagation. But what if the parent plant is infected with a virus? No worries. There's a small part of the plant called the meristem, present at the tips of shoots and roots, which usually stays virus-free. Scientists can extract this meristem, grow it in vitro using tissue culture, and produce healthy, virus-free plants. This method has been successfully used in banana, sugarcane, and potato cultivation. Here's something even more futuristic: somatic hybridization. Scientists can isolate single cells from two different plants. After removing their cell walls, they get protoplasts, cells without walls, just surrounded by a plasma membrane. These protoplasts can be fused together in a lab, forming a hybrid protoplast. This fused cell is then grown into a full plant, carrying traits from both parent plants. An example? Scientists once fused tomato and potato protoplasts to create a plant called Pomato, a plant that produced tomatoes above the ground and potatoes underground. Although it wasn't commercially viable, it showed what's possible when plant biotechnology meets creativity. So, from cloning plants in test tubes to curing viral infections and even creating weird hybrids like Pomato, tissue culture is one of the most powerful tools in modern agriculture and plant science. It not only saves time, but also ensures we have healthy, identical, and high-yielding plants ready to feed a growing world population. If you found this video informative and fascinating, don't forget, your one like, share, and subscribe will help us create more valuable content. Stay curious, stay informed, only on Knowledge Chronicle.